Today's class is about neutral evolution. We're going to go through what we mean by that, so the definition. We're going to talk about something called the Genotype-Phenotype map. And then we're going to think about why you can have variation with no fitness change. This is very relevant for neutral evolution. The importance of the environment. And then I'm going to focus a little bit on this model of genetic drift called the gene pool model, and talk about founder and bottleneck effects, which are these important genetic effects of related to neutral evolution. So this is related to this, one of the variants of this most important sentence that we keep seeing. If there's no variation in reproductive success, if there is variation in reproductive success, if there's variation in the trait, and if there's weak or no correlation between the trait and reproductive success, so this is where this differs from adaptive evolution. If that is the case and there's inheritance of the trait, then there will be neutral evolution. So because the relationship between the trait and reproductive success is this statistical relationship, you can have strong adaptive or weak adaptive, or the amount of adaptive evolution relative to neutral evolution depends on the strength of this relationship. And that will place the evolution of this trait somewhere on this spectrum between neutral and adaptive. But we're going to focus mainly today on when it's strongly neutral evolution. So it's useful and it's important to understand before we go on about this concept of the genotype-phenotype map. And the idea with this is that there are these three layers: the genotype, so what are your genes? What's your DNA? The phenotype, so what is your realized phenotype that's determined at least partially by the genotype, but also by the environment. And then your fitness, which is a result of your phenotype. So the genotype affects phenotype, which affects fitness. And as is quite well illustrated on this diagram, I think, you should realize that different genotypes can result in the same phenotype. So G1 and G2 here both result in phenotype P1, for example. We'll come on to how that's possible in a minute. And then different phenotypes can result in the same fitness. So P1, phenotype P1 and P2 result in fitness F1 in this schema here. So it's not quite as simple, or the world is not quite as simple as different genotypes having different fitnesses. There's this layer in between of the phenotype, which makes things slightly more complicated. One of the best examples illustrating this kind of concept is the famous experiment by Richard Lenski, which has been running in the US, in Minnesota, I think, on E. coli. So, apart from causing stomach upsets every now and again, it's a lab species. It's studied quite intensively, and his team have been following populations of this, the same population, largely from 1988 through to now. They've tracked this. Well, this picture is from 2 or 3 years ago now, but that's the number of generations that have been followed in the lab. So 63,000 generations of E. coli population dynamics. And what they're doing is studying this genotype-phenotype-fitness map in various ways. And their main findings can be summarized like this. Well, evolution is real, it's happening. And in several of their experiments, they're looking at how E. coli respond to different environments. So they have an environment that's been enriched with glucose. And they have tracked how these E. coli have altered via evolution, via adaptive evolution, to become fitter in this environment. So they've optimized themselves for this sugar-rich environment. So that's adaptive evolution. And they show that this is replicatable in different lines, different experiments. But what they have shown, is that the exact genetic and physiological pathways that these different lines have taken to get this increased fitness have varied. So you've got different genes producing the same fitness outcomes. So the key take-home message relevant to this class is that both adaptive and neutral evolution are happening simultaneously in this population, and that's the case everywhere. So you might now be wondering, well, how can genes vary but not fitness? And these are the main reasons. You've got different physiological pathways that can lead to the same outcome. You've got something called synonymous genetic substitution and synonymous amino acid substitution in proteins, and something called canalisation. So I'm going to dig into each one of those now. So synonymous substitution. So your genetic instructions in your DNA take the form of codons, which are 3-letter sections of code that each one encoding for an amino acid. So this is the instructions for the building blocks of proteins. So the amino acids make the proteins for your enzymes and so on. And these codons are the instructions. So for example, CGA will make arginine. And a typical gene will have lots and lots of these codons. So it'll have a start codon that says gene starts here, then it'll have the instructions, and then it'll have a codon which says that's it, you've finished. Amongst this, you also have some non-coding bits as well, called introns. So here's an example of synonymous substitution. So the amino acid leucine is coded for by these 6 different codons. So they're all different genetically. These are genetically different codons, but they all result in the same amino acid. So a mutation that takes the codon from being AAT to AAC has no important effect in terms of which amino acid is produced. But that's still genetic variation. Okay? So genetic variation can result in the same instruction in terms of which amino acid should be produced. And you get similar kinds of substitution effects in proteins. So amino acids, they vary, but they can have analogous properties. So for example, in a binding site for a protein, you might need to have hydrophobic. The site might need to be hydrophobic. So as long as there is a hydrophobic, so water-fearing amino acid in that position in the instructions, that enzyme will still function. So you can have some change of amino acids, but not change the function of the protein, the enzyme. The final way that you can have variation, but no change in function, is via this process that's called canalisation, which is, you can think of it as being developmental stability. And this canalisation is literally from the word canal. You know, you're constraining where water can go by channeling it through a canal. These were first put forward. This idea was first put forward by this guy Waddington in 1942, who said developmental reactions as they occur in organisms submitted to natural selection are adjusted so as to bring about one definite end result, regardless of minor variations in the conditions during the course of the reaction. So it's saying that you can have that the physiology tolerates quite a bit of variation and still results in some in the same end result. A good example of this, I think, is the homeostasis of body temperature. So you can have quite a bit of variation among humans, for example, or any other mammal species, I guess. But our body temperatures are really constrained. You know, there's a little bit of variation in what our body temperature is, but it's pretty constrained. And the idea is that it's constrained by this canalisation process. And what you would need, so you've got all this sort of hidden variation that's being, the genetic variation is there, but the result is canalised. So you've got a lot of variation, but a very constant end result. And the idea is that you would only see an expression of this variation under really extreme circumstances. So the canal only overflows under very special circumstances. So canalisation is when traits show very little phenotypic variation, despite the fact that they have that they can have underlying large amounts of genetic variation. What causes that? Well, it varies depending on the kind of trait that you're interested in or that you're thinking of, but the idea is that it's due to buffering. So these kind of traits are usually determined by many complex interacting physiological and genetic processes that are constraining each other so as to have this fairly fixed end result. And this has relevance to neutral evolution because if that variation isn't expressed, then in the phenotype, then that variation is evolving via neutral evolution. There's no selection pressure to constrain that variation. It's there and it can vary, but there's no variation in fitness. So when thinking about neutral evolution and adaptive evolution as well, the environment in which the species or the population finds itself is key. So in one environment, a trait might be evolving neutrally, and in another environment, it might be evolving adaptively. So a classic example of that, or a very clear example of that, is antibiotic resistance. So I showed you that video last time of this antibiotic resistance developing on this mega plate of agar. In that environment, when the bacteria is under strong selection to survive in this hostile environment, that's when there's adaptive evolution happening. You know, it's being pushed by selection. But if there was no antibiotics in its environment, there's still variation, and those genes that are there, the standing variation, is going to change through time without selection pressure. So evolution is still happening, but it's happening neutrally. So there's evolution is simply the changes in gene frequency through time. So as long as there's variation there will be changes in gene frequency via random processes. So environment is key. So if these processes are random, can they cause evolutionary change? Like I just said, evolution is just simply defined as a change in inherited traits or gene frequencies in populations through time. So although neutral variation doesn't influence fitness, it won't have a favourite, it can change through time. So you might expect this to lead to some sort of equilibrium where there's no change on average through time. Let's illustrate that. And that's true, and it can be proven mathematically that it's true, but it only is the case when the populations are infinitely large, and populations are not infinitely large. They're usually quite a lot smaller than infinitely large. So, although mathematically these sort of random choices that are being, random selections that are being made, this neutral selection, you would get sometimes just by chance a series of positive changes and a series of negative changes, and these will not balance, or could potentially not balance as you go through time. And this is what leads to this process called genetic drift. I'm going to illustrate this now. So genetic drift is also known as, is driven by this process that is known as the Mendelian lottery. So the transmission of your genes to your offspring is a random process. It's a 50-50 chance whether you inherit a chromosome from your mother or your father. And you can see, so it's the sex chromosomes, for example, there's a 50-50 chance that your offspring will be, for each one, will be a male or a female, right? But everyone knows families where they've got a number of children and they're all boys or they're all girls. That's just chance. So if you have a population that's quite small, then you could end up with a skew just by chance of sex ratio. But with a large population, you wouldn't expect that. So if women had 100 children, it would be extremely unlikely that all of them would be male or all of them would be female. But if they only have 2 or 3, then it actually becomes quite likely that just by chance they have all males or all females. This is the essence of this Mendelian lottery thing. So this kind of variation is very important for small populations and less important for big populations, this genetic drift. And this is thought about and modeled mathematically by this, what's called the gene pool model, which models the genes in the population as like a pool, so illustrated by that filled bottle of yellow and red balls, yellow and red genes. So these parents contribute their genetic material to this pool, is the idea, and then their offspring, according to this mathematical model, the genetics of the offspring are a product of random choices from this pool. So this has a lot of assumptions, one of them being that mating is random, so you're randomly picking from this pool. Mating is not normally random, so this is a simplification. I've made a simulation of this, which you should be able to get to if you follow this link here. But this is what it shows. You can play with it later. So this, on the horizontal axis, is time in terms of generations, so generation time. and on the vertical axis is an allele frequency. So this is genetic material. And the idea is that there's two alleles, A1 and A2, and this just shows A1, and the frequency, the amount of A2 will be 1 minus that. So I started off this population with a 50-50 mix there at the left-hand side. And then what I've done is I've simulated, I think there's 20 simulations of the population. So every population is starting off with the same mix of alleles, 50-50. And there's some movement around this mean, right, around this initial allele frequency. So there's a line that goes down, for example. This is just by chance having the random process is resulting in a reduction in the frequency of allele A1. And then there's other populations where it's sort of more fluctuating around the middle. So the big idea here is that there's no selection happening. So if there was selection, it would be forcing allele frequency to be held at whatever the optimum is. Okay? So this is why it's this spread is symmetrical, because there's no better solution. Fitness is staying the same. It's just random processes that are resulting in change through time. So that's with a large population. If we shrink the population, theoretically, we would expect these random processes to become more important. In the same way that you would expect, it wouldn't be unusual to have a woman with 3 boys, but it would be unusual to have a woman with 100 boys. Even more so than, well, for a number of reasons, but you know what I mean. So here is what it looks like with 100, with a much smaller population. I forget how many I've fixed the population to. But you can see there's wild fluctuations here. You can see these random processes are becoming very, very important, and including in some cases, like the one on the top right there, after 40 or something like that, 42 generations, this run has, the allele frequency has become fixed at 100% allele A1. So in this population, genetic diversity is now shrunk. So you can imagine this going on for a whole sets of genes. This is just working with this one gene with 2 alleles. And this is where we relate it to the cheetah example that we saw the video of. When the population is quite small, like many species of conservation concern, you can get fixation of alleles and reduction in genetic diversity that's driven not by pressure, selection pressure, but by random chance. So there is a much greater chance of randomly reducing genetic diversity in a small population than there is in a large population. So some other consequences of this kind of model, this kind of thinking. One of these is the founder effect. And this is, you start off with a large population, and a founder effect is when you have a small chunk of that population migrating to a new area and starting afresh in that region. So you get that when you have migration events, like in human history, you've got the ancestral home of humans in Africa there, and these migration events are not huge groups of individuals moving into new areas, but small groups. So you get founder effects where those small groups, just by chance, have a different genetic makeup than the parent population. And that's why there's variation, genetic variation, among humans if you go to different places. There's a really nice example of that that was published last year, where scientists were looking at the evolution of birth canals in women. And the old idea was that the birth canal, the shape of the birth canal is an adaptive, is the result of adaptive natural selection. So there's these opposing forces that are driving this shape. So it needs to be wide enough that it allows babies with big heads and big brains to pass through, but it needs to be narrow enough to allow efficient walking. If you think about our ancient ancestral times, we were not able to ride on our bikes or take taxis or ride in cars. We were walking around and gathering and hunting. So it's important to be able to move around. So the old idea is that there's these two adaptive forces that are driving the shape of the birth canal. And if that's the case, then what you would expect is globally, in all the populations, these forces are the same. Right? People are people everywhere. They do the same kind of things. So you would expect a consistent shape in all these different areas. So these guys, they went and they measured old skeletal remains using rubber bands and measuring tapes. And what they found was that there was the variation within populations, so variation in Europe, variation in Asia. Within those populations, the amount of variation declined as you moved away from the birthplace of humans. And this is consistent with the idea that every time human populations move on to a different place, it's a subset of the ancestral population of reduced genetic diversity. So this is a statistical sampling type of thing going on. And they found that phenotypic difference correlated with genetic distance. So by phenotypic difference, I mean difference in width, height of the birth canal, and genetic distance, I mean how distantly related these populations are. So what this suggests altogether is that this variation in this really important trait for humans is driven largely by chance. So chance, these founding effects that are neutral evolution type effects, have shaped our birth canals, and not selection. So that is the illustration of, on the top graph, it's on the horizontal axis, it's distance from central sub-Saharan Africa, and on the y-axis it's phenotypic variation. So it's declining as you move away. So this is the progressive smaller and smaller samples that are being that are founding these new populations. And then on the bottom graph it's genetic distance, so how distantly related are points from each other, and phenotypic distance. So as points get further genetically more distant from each other, relative, how distantly related pairs of points are, they're also more phenotypically different. So the shape is changing as well as genetic distance. A related principle is this bottleneck effect. And the idea is that it's this sample of individuals, so that the parental population is represented here by the beads, or are they jelly babies, I'm not sure, in these bottles. And this sample of individuals are coming out through the bottleneck, hence the bottleneck effect. So the bottleneck and the founding effect are very closely related, actually. So you can imagine, in this case, shaking out some of these, small number of these different colored beads into a container. And if you take a small sample, it's going to be quite likely to get all one color, or not at least not unlikely. And if you shake out a large sample, so it's not such a big bottleneck, then it's quite unlikely that you will get this. So this bottleneck effect, the bottleneck is a kind of constraint on genetic diversity that's resulting. And one of the key examples of this is in the cheetah. The idea is that 100,000 years ago there was a founding effect from the ancestral population of cheetahs, so you've got a restriction of genetic diversity then, and then you have a bottleneck effect that occurred about 10 to 12,000 years ago, which is driven by human hunting. There's this theory, delightfully named theory, the human overkill theory, for extinction of large species like this. They almost drove the cheetah to extinction. The cheetah was driven to extinction in North America. So you end up with this small genetic sample of, so the cheetahs that exist now have passed through this bottleneck. They are a genetically quite homogeneous sample of the parental population, just because of this sampling effect. And because of this, they are fairly inbred. They don't have so much genetic variation, and they suffer apparently from poor sperm quality and poor resistance to disease, which has obvious conservation implications. So those are some conservation-oriented discussion points, I guess. Neutral variation also has some really, really useful applications. So certain parts of your genome, or human genome, is evolving neutrally. And we can use that neutral variation to get information about evolution. So the key thing that I'm thinking of here is to figure out relatedness of different species. So how different are these sequences in these neutrally evolving sequences? How different are they for different species? This needs to be... So the idea is that if something is evolving neutrally, if the genome is evolving neutrally, or the part of the genome is evolving neutrally, then how different they are in different species tells you how long they've been separated, because this is it's a random process, so they're going to diverge on average the more time that there has been. So these are the kind of parts of the genome that are telling us, that are giving us genetic information on distance of relationship and date for when we diverge from a common ancestor, for example. So this is used for phylogeny construction. Another example is genetic fingerprinting. So if our genotype was under extremely strong selection and there was no neutrally evolving parts of it, then all of our genotypes would be extremely similar, and then you wouldn't be able to do genotyping, because I would be quite likely to share genotypes with a few of you. So you would not be able to tell who had murdered my neighbour, for example. So it's useful for forensics. And then thinking more about the phenotype, the trait level, traits like fingerprints and iris structure, these are not under selection, and that's why they vary so much. So any trait that varies hugely, like fingerprints and iris structure, these are the kind of traits that are that are neutral, that are neutrally evolving. They're not under selection, and that's what makes them useful for ID. So I've given you a very broad brushstrokes picture of neutral evolution. The definition, this really important concept of the Genotype-Phenotype-Fitness map. I've talked about why you can get variation with no with no fitness change via these synonymized genetic material or protein sequences. I've mentioned that environment is key, so whether a trait is evolving adaptively or neutrally depends on the environment that it's in. I've mentioned this gene pool model of genetic drift. This is the way that evolutionary biologists think about this type of evolution, with the key processes there being the founder and the bottleneck effect. Right, thank you very much.